Let's start with glycolysis. Today I am going to teach you about glycolysis. I will teach you first the very brief intro of glycolysis, then I will give details of each step individually. This will be a very long video, so if you're looking for a very short video containing less amount of knowledge, then you can find another video. This video is not for you. This video contains all the knowledge that you should know at undergraduate level. That is why this video is long. So let's start. First is what is glycolysis. As I told you, a brief intro. So what is glycolysis? It is a metabolic pathway of breakdown of glucose into two molecules of pyruvate. Here I wrote down the reaction. You can see glucose is being undergoing cascade of reactions. That cascade of reaction is actually metabolic pathway called glycolysis. So being finally converted into two molecules of pyruvate. Next point, principal pathway of carbohydrate metabolism. For glucose metabolism, glucose must undergo glycolysis because it is the first or principal or pathway for glucose metabolism, because glucose must undergo glycolysis so it can be used for energy. Occurs in all cells of the body, but brain and RBCs, they require glucose as their main energy source because 80% of brain's energy come from glucose metabolism, and RBCs cannot use aerobic respiration, that is aerobic metabolisms. As a result, it is totally depending on the glycolysis, anaerobic glycolysis actually, to reach its metabolic or needs. So both occurs in both aerobic and anaerobic conditions. Why? Because glycolysis does not need oxygen. Although it is an oxidative reaction, although it's a catabolic pathway, but it does not require oxygen. So it occurs in both aerobic and anaerobic condition. That is why it is most important pathway. As I told you, anaerobically occurs in RBCs. It occurs in muscles anaerobically when muscles are under extreme stress or has been used for hours, that is extreme exercise. Then muscles enter hypoxic states at which they use glycolysis anaerobically. In cancer cells, glycolysis always occur anaerobically because the rate of growth of cancer cells is extreme. So a cytosolic pathway, and all the enzymes involved are cytosolic. Glycolysis occur in cytoplasm, so even so enzymes that are used will also be present in the cytoplasm. So let's go to the first step. Step 1 is conversion of glucose into glucose 6-phosphate. I write this down, this reaction here. Why this reaction occurs? First step of glycolysis is to make sure that glucose that has entered into the cell to undergo glycolysis does not go out of the cell again. So to make sure it does not go out of the cell, we phosphorylate the glucose to make it polar and large molecule, so it cannot go through the membrane now. So now it is committed to undergo glycolysis. The enzyme involved in this reaction is hexokinase or glucokinase. You see, hexokinase, I write this down in equation. The glucokinase and hexokinase both are different enzymes. Both are actually isoenzymes performing the same functions, but they are present in different cells. Some cells have hexokinase, while some cells have glucokinase. Why? I will tell you later. What is actually, this is a cytosolic enzyme, like I told you. What these are actually kinases. Kinases are enzymes associated with phosphate group transfer. What actually happens in hexokinase, strips a glucose from a group and gives it strips a phosphate from group and gives it to glucose, forming glucose 6-phosphate. But the question is, where does that phosphate come from? Well, the answer is ATP. And ATP is consumed, that is, glucokinase strips off phosphate from ATP and gives it to glucose, forming glucose 6-phosphate. Hence, this is a phosphorylation reaction. This is an irreversible reaction, hence a regulatory point. All the irreversible reactions in metabolic pathways are the reactions which undergo regulation, that is allosteric regulation and hormonal control. Hence, this reaction is very much important from regulatory point of view. Now, what will be actually regulated? Well, these enzymes which I just told you, glucokinase and hexokinase, will be the regulated. So what actually happens, that enzyme is negatively regulated by its end product. Here glucokinase and hexokinase end product is glucose 6-phosphate. Glucose 6-phosphate what actually does, it negatively feedbacks to these enzymes. That is, if glucose 6-phosphate concentration is greater in the cell, glucokinase and hexokinases will not work. But if glucose 6-phosphate concentration is very low in the cell, then glucokinase and hexokinase, they work very much actively. So this is the negative feedback mechanism. Well, hexokinase versus glucokinase, I wrote down these differences, but to explain them, first we need to know what Km is. Km is the concentration of substrate at which enzyme achieves half Vmax. If we in this case, glucokinase or hexokinase's Km will be will be actually the concentration of glucose at which they achieve half of their Vmax, that is their maximum velocity. So here I draw a graph, Vmax versus glucose concentration. As you can see, hexokinase, we increase the glucose concentration very less, and hexokinase reaches half of its maximum velocity. But then it grows and finally it reaches a plateau state. That is, it reaches its Vmax. But this Vmax is low as compared to glucokinase. Glucokinase has very high Vmax, but as compared to hexokinase, it requires large concentration of glucose to reach half of its Vmax. That is why the Km of glucokinase is low, Km of hexokinase is high, but Vmax of glucokinase is high, Vmax of hexokinase is low. We know that Km and affinity to substrate are inversely proportional. That's why affinity of hexokinase to glucose is high, whereas affinity of glucokinase to glucose is very much low. These three differences I explained you, but the fourth difference is that all cells contain hexokinase, but only liver and beta cells of pancreas contain glucokinase. Why? Well, the answer is very simple. In all cells of our body receive moderate concentration of glucose that is usually low. Well, because the Km of glucokinase is very high, glucokinase cannot work in low glucose concentration. But because Km of hexokinase is low, that is why hexokinase can work in low glucose concentration. That's why in all cells of our body we have hexokinase. But what about glucokinase? Glucokinase is in liver. So why glucokinase is present in liver? Because liver of our is organ of our body which receives all the glucose coming from GIT. So liver cells receive very large quantities of glucose. As a result, the hexokinase has very low Vmax. That is why hexokinase cannot handle this large quantity of glucose and phosphorylate it all for glucose 6-phosphate. That is why we have glucokinase there. Glucokinase has very high Vmax, so all the glucose that comes to liver cells can be phosphorylated to glucose 6-phosphate. These two differences I wrote down in the last 2 points. Another important point, well, it is a misnomer, very famous misnomer, very popular misnomer, that glucokinase is specific to glucose as compared to hexokinase, which is not specific to anything. Well, actually, both enzymes are not specific to anything. These both enzymes work in all hexose sugars. It was thought that glucose only works for glucose, but that's not true. Glucokinase and hexokinase, they both act on all the hexose molecules. Well, another important thing you should know about are the glucokinase actions, which are outside the phosphorylation. You see, in beta islet cells of pancreas, I told you the glucokinase is present, but I didn't tell you why. Well, the answer is beta cells of pancreas, it causes sensitivity to glucose level and release of insulin. Well, this point is self-explanatory, but I will explain to you how does that happen. Well, increased glucose in blood causes increased phosphorylation that glucokinase is capable of, and hence increased ATP production into the cell. What next happens, that increased ATP production causes blockage of ATP potassium pump. As a result, the voltage changes take place. These voltage changes called cause voltage gated calcium channel to open. As a result, calcium influx occurs, Hence, calcium binding to insulin vesicles, vesicle binding to plasma membrane and releasing insulin. Well, this point I need to explain. Beta islet cells of pancreas contain insulin. That insulin is actually inside of vesicles inside its cell. So what happens that calcium when comes into the cell, the calcium bind to those vesicles and force them to fuse with the plasma membrane. As a result, insulin is released. So glucokinase is indirectly actually causing insulin to be released. Same way, it is also sensitive or monitors glucose level in hypothalamus. Well, the second point is in liver phosphorylates all glucose coming to it. As I told you before, that liver receives high quantities of glucose, so glucokinase converts it all to glucose 6-phosphate. This was step one. Step one was only this. All of this, all the rest of the stuff I told you was actually the extra information that you should know at undergraduate level. So this is a part one. I end my part one here. In the next part, I will teach you the step two.